This video is a long-form narrated commentary that sits alongside part 7 of my GCSE Science Sprint series. So here we're going to go through exactly the same exam question, but just talking about why we're writing what we're writing. This question typically appears as an extended response question, but depending on how easy or challenging the exam board want to make it, they could give you a label diagram to help you to formulate your answer, or they could give you a diagram that you need to label before you start, or they could even extend the question a little bit further by asking you about how these different tissues are specialised in order to allow them to perform their function. So to begin with, when describing the structures in the leaf, you're probably going to start with something that isn't in fact a tissue because it's not made of living cells, and that's the waxy cuticle. Underneath that waxy layer, which helps to prevent dehydration and water loss, you've got your first actual tissue, and this is your upper epidermis or upper epidermal layer. And then underneath that is your palisade mesophyll, followed by your spongy mesophyll, and integrated in that spongy mesophyll you have these vascular bundles made of xylem and phloem. And then finally at the bottom of the leaf you have another epidermal layer, and this contains the majority of the stomata, which are surrounded by guard cells. You do get a few stomata in the upper epidermal layer, but far fewer of them, because obviously if you have those stomata at the top, you're more likely to be losing water, and the plant is just running out of water through transpiration. So this waxy cuticle layer is there to reduce water loss. The epidermal layers are there kind of like a skin. They're basically going to cover the outside of the leaf. And then the palisade mesophyll is the main site of photosynthesis. You do have photosynthesis happening in some other tissues, but that's primarily where it's going to happen. The spongy mesophyll is for gas exchange, so the exchange of carbon dioxide going into those cells to be used in photosynthesis and the oxygen coming out as a waste product. The xylem and the phloem are for transport, so the xylem is transporting water and then also minerals dissolved in that water, and the phloem is transporting sugar. One important thing to note here is that it's not actually glucose that it's transporting. That glucose is converted into a different model before translocation takes place. And then this lower epidermis containing these guard cells, well, the epidermis is just another covering layer, but the guard cells are there to open and close the stomata in order to control water loss and transpiration. So as the plant becomes more dehydrated, those guard cells are going to become flaccid, causing the stomata to close, and that shuts down transpiration and prevents the plant from losing more water. But obviously that does have an impact on its ability to do photosynthesis. Then, as we said, it's possible that you could be given a question where they actually ask you to extend this and not just say about what the functions of the tissues are, but how they're adapted to do these. So we can talk about the fact that this upper epidermal layer, contains very few chloroplasts, or maybe no chloroplasts at all, to allow light to penetrate down to the palisade mesophyll layer. And that, of course, is packed full of chloroplasts for maximum photosynthesis. And also not really a specialisation of the cell, but just thinking about how the leaf is made up. Those palisade mesophyll cells are right near the top of the leaf to maximise light absorption. Then the spongy mesophyll has all these gaps to allow for the gas exchange. And then in terms of the individual vessels within that vascular bundle, the xylem are these hollow, woody, dead tubes with no divisions in between what were originally cells, so the water can just flow freely all the way up the plant along the transpiration stream from the roots up to the leaves and out. Whereas the phloem, in contrast, they are living cells, so you have these sieve tube elements with a companion cell next to them that's sort of supplying them with energy and things, so that the actual sieve tube itself can have minimal other subcellular structures in and just give maximum space for translocation to occur. Thank you very much for watching, and I hope you're finding this series of videos useful in your GCSE Science revision. If you are enjoying them, then don't forget to like, subscribe, comment and share, and I'll see you soon for some more GCSE Science revision videos.